histology of lymphatic system, if you remember, once I have explained it in the class, but I have not shown you the slide. This already I have explained, so this will be a revision class. Okay. So here directly we will discuss the microscopic structure of spleen, then microscopic structure of lymph node, and microscopic structure of thymus. Only these three structures. Tonsil already we have discussed, it is done. Okay. Now, what do you know about the spleen? You know that spleen is a intraperitoneal organ, right? What else do you know? Now, before going to the spleen, let me tell you one thing. Here, what will be the concept of the structure? जितना भी structure है, for example, spleen, lymph node, thymus, the concept is, first of all, capsule will be there. From capsule, the trabeculae or the septa is going inside the gland and it is dividing into lobules. And through the capsule or septa, blood vessels, other structures, nerves, they are passing. Okay, and but it depends in each and every lymphoid organ, those things are not there. Next, you will see that stroma is there and parenchyma will be there. Stroma means जो connective tissue capsule होगा, trabeculae होगा, blood vessels, those are the stroma. And another important part in the stroma is reticular fibers and reticular cells. They are forming the framework of any lymphoid organ. So that will come in the stroma. Next is the parenchyma. Okay? Parenchyma, you will see that two things are there in the parenchyma. One is cortex and one is medulla. Okay, spleen में थोड़ा अलग रहता है, बाकी दोनों में cortex medulla रहता है. Okay, so you have to know the differences between the cortex and medulla, how it appears. Okay, so spleen, as you know, it is the largest lymphoid organ. Okay, उसका main function क्या है? Filtration of the blood. How it filters the blood? Not like the kidney. Then how it filters the blood? Mainly it removes the damaged or old RBC which are present in the blood. That is the filtration done by the spleen. Next, during intrauterine life, spleen acts as a hemopoietic organ. That means formation of the blood cells are taking place in the spleen during intrauterine life. Okay. Next, histologically, if you see, first of all, you will be getting a capsule, then a trabeculae. From capsule, trabeculae is going into the substance of the spleen, and then white pulp and red pulp. So the parenchyma of the spleen is mainly made up of red pulp and white pulp. Okay, this is the things which we are going to see under microscope. So this is the microscopic structure of spleen. So you have to understand the structure. First of all, spleen is having connective tissue capsule. Okay. So can anyone tell me the connective tissue spleen is an intraperitoneal organ. That means it is covered by peritoneum. Now what about the capsule? Where the capsule will be present? Outside the peritoneal layer or inside the peritoneal layer? Inside the peritoneal layer. Okay, that means deep to the peritoneal layer, serous layer, the capsule is present. Okay. Now capsule, you can see it is formed by dense connective tissue. Myofibroblast cells are there, and from there the trabeculae arises. Okay, and they will go deep into the parenchyma of the spleen. Next, if you see the parenchyma, it is made up of white pulp and it is made up of red pulp. Okay, white pulp doesn't mean that color is white. Okay, it is a feature, or you can say important identifying feature of spleen, red pulp and white pulp. Okay, so red pulp में क्या रहता है, white pulp में क्या रहता है, that you have to know. So if you see the white pulp, what is the appearance? Darkly stained. We are studying histology, so we should know which part is darkly stained and which part is lightly stained, right? तो ये जो white pulp होता है, what is the appearance? Darkly stained, that also basophilic. तो color कैसा होगा? Yes, B for B, basophilic मतलब B, blue also B. So ऐसे याद रखो, B for B, basophilic is B, blue is also B. तो if it is basophilic, it will be blue in color. ठीक है? Next, in case of white pulp, first of all, it is a darkly stained, basophilic in appearance, and then a central arteriole is present. Okay. Now this particular arteriole will be covered by two different types of lymphocytes. Okay. Now in first case, or in most of the cases, it is covered by periarterial lymphatic sheath, or also called as PALS, periarterial lymphatic sheath. In this particular area, the central arteriole is covered by which type of lymphocytes? T lymphocytes. Okay, it is covered by T lymphocytes. Next, there are some areas where B lymphocytes proliferate, okay, and then it will cover the central arteriole. Now, those are called as splenic nodules or Malpighian corpuscles, where instead of T cells, you will be getting the B cells or B lymphocytes, okay. Now, in this particular case, a germinal center will be there, okay. In this case, a germinal center will be there, and due to the presence of germinal center, the location or the position of the arteriole will be eccentric. What is the meaning of concentric and eccentric? Or eccentric means? Concentric means it will be present in the center. Eccentric means towards the periphery. Okay? Eccentric means towards the periphery, not at the center of the circle, towards the periphery of the circle. That is the eccentric. Okay? Now you have to understand one thing. Focus over here. तो जो white pulp होता है, first of all, it is darkly stained, it is basophilic in nature. ठीक है? Now this particular part is having a central arteriole, and this central arteriole is surrounded by two different types of lymphocytes. If it is surrounded by T lymphocytes, then it is called as periarterial lymphatic sheath or PALS, as simple as that. Instead of T lymphocyte, if it is covered by B lymphocytes, then it is called as splenic nodules or Malpighian corpuscles. ठीक है? In this case, it is concentric in nature. मतलब जो arteriole रहता है वो center में रहता है, ठीक है? But in this case, due to the presence of germinal center, the central arteriole is pushed towards the periphery. That's why it is eccentric arteriole. Okay? Now, can anyone tell me what is this germinal center? See, there are some cases where the lymphatic nodule, okay, is called primary lymphatic nodules are there and secondary lymphatic nodules are there. Okay. Now, in secondary lymphatic nodules, germinal centers are present. Okay, germinal centers are present only because if that particular organ is exposed to any antigen. Okay. In general, it will be T lymphocyte. If the spleen is exposed to any antigen, at that time germinal center will appear, where instead of T lymphocyte, you will be getting the B lymphocyte. Germinal center, if you see the appearance, the center part, okay, will be brighter in appearance, and peripheral part will be dark in appearance. Why? Because at the center part, the cells which are forming, they are immature lymphocytes. Okay, that is the appearance of germinal center. Next, so this is the white pulp. Now, what is the red pulp? What is the appearance of red pulp? कैसे दिखता है? Brightly stained. That also eosinophilic. ठीक है? तो अगर eosinophilic हुआ तो what will be the color? Pink color. Okay. So here you can see lightly stained and it is eosinophilic. So like this you can easily differentiate between the white pulp and red pulp under microscope. ठीक है? Next, what about the red pulp? How it is formed? It is formed by two things. As you can see, cords of Billroth. Now, what is this cords of Billroth? Different types of cells which are present in the red pulp. They are in the form of anastomosing cords. What are the different types of cells? RBCs, lymphocytes, macrophages, plasma cells. Okay, like these different types of cells are there, and they are arranged in the form of branching and anastomosing cords. And those cords are separated from each other by sinusoids, and those are called as splenic sinusoids. Okay, that's why when you will see the red pulp, some empty spaces can be seen, small empty spaces can be seen. Okay, those empty spaces are nothing but the splenic sinusoids. Now, splenic sinusoids में क्या रहता है? What do you think? What will be present in the splenic sinusoids? Sinusoids में क्या रहता है? Endothelium रहता है. Okay, sinusoids are lined by endothelium. Okay, and in most of the cases, their basement membrane are incomplete. Okay, basement membrane complete नहीं होता है. Gaps are there in the basement membrane. Now, here some modifications are there. The sinusoidal cells, okay, they are elongated in one direction, and if you see the shape, it is banana shaped. Okay, it is banana shaped, and they are called as stave cell. STAVE, stave cells. ठीक है? तो ये क्यों रहता है? Why they are present? Now these particular stave cells, they will control the opening and closing of the sinusoids. As a result, they will control the flow of blood through the sinusoids. Now, if any damaged RBC or if any older RBCs are passing through this sinusoid, they will not allow it to pass through the sinusoid, and the filtration is taking place over here. Okay, so as you can see, line by elongated endothelial stab cell filled with RBC. So this particular part makes the filtration. Okay, now in between the red pulp and white pulp, an area is there that is called as marginal zone. रेड पल्प एंड वाइट पल्प के बीच में एक एरिया है दैट इज कॉल्ड एज मार्जिनल जोन. इसमें क्या रहता है? एंटीजन प्रेजेंटिंग सेल्स. ओके, दैट पर्टिकुलर एरिया इज formed by antigen presenting cells. Okay, so that is the overall structure of the spleen. Now I will show you the diagrams. You have to identify the parts. So whatever we have discussed, everything is mentioned, just go through it, you will understand. Okay, now see this particular diagram is very important. So in case of white pulp, I have mentioned that first of all periarterial lymphatic sheath is present. Okay, and what is there? Malpighian corpuscles. Okay, now here the Malpighian corpuscles will be formed. Now in case of periarterial lymphatic sheath, just see this is the central arteriole. Around the central arteriole you can see the aggregation of lymphocytes are taking place. So this will be T lymphocytes. ठीक है. Now if you take a section, what will be the appearance? यहाँ पे अगर ऐसे का मान लो एक सेक्शन दिया TS, तो कैसे appearance क्या होगा? So a round section will be there. Center में क्या रहेगा? Arteriole will be there in the center. Or around the arteriole, what you will see? T lymphocytes. ठीक है? So this is the periarterial lymphatic sheath. Next, here instead of T lymphocytes, B lymphocytes are proliferating. Okay, and due to the presence of the germinal center, you can see the central arteriole is pushed towards the periphery. Now, if you take a TS, what you will see? Same thing. They will be eccentric in nature. Okay, so this is the appearance between the the periarterial lymphatic sheath and Malpighian corpuscles. So this is the microscopic structure of spleen. Now in the microscopic structure of spleen, first of all, what is this? ये क्या है? Capsule. As simple as that. Capsule will be there. Now from the capsule, can you see the trabeculae is extending into the substance of the spleen? Yes. Now through the trabeculae you can see some blood vessels. Can you see the blood vessels? Okay. So these blood vessels are also coming in. Okay, through the trabeculae. Next, what you have to identify in the spleen? Red pulp and white pulp. Can you identify where is white pulp? यहाँ पे white pulp कहाँ पे है? Anyone please come and show me where is the white pulp? Where is the white pulp? हाँ, these are the white pulp. This is one white pulp. This is white pulp. This is white pulp. This is white pulp. All these are white pulps. ठीक है? Why it is white? What is the appearance of the white pulp? Darkly stained. That also basophilic. That means blue color appearance. Just see, these are basophilic blue color appearance are there. Now, by looking at the white pulp, can you tell me where is the location of arteriole in most of the cases? Eccentric. ठीक है? Eccentric है. Now, if it is eccentric, then see this is the germinal center. ये देखो, this is the germinal center. This is the germinal center. This is the germinal center. Okay? तो if you see the germinal center, उसका जो appearance है, it is lightly stained because in that particular part newly formed or immature lymphocytes are present, and at the periphery you can see it is darkly stained. See this area is darkly stained, this is lightly stained. Okay, so this is the appearance of white pulp. Okay, next where is the red pulp? Red pulp कहाँ पे है? ये देखो, this is the red pulp, this is red pulp. ठीक है, red pulp में क्या क्या रहता है? Two things are there in the red pulp. First of all, what is the appearance of the red pulp? First of all, it is lightly stained and eosinophilic. Okay, तो pink color का appearance होगा. Next, it is made up of two things: splenic cords of Billroth and splenic sinusoids. तो sinusoid का appearance कैसा होगा? Blank spaces. जो sinusoids होता है, how they appear, blank space. So you can see so many blank spaces are present, small blank spaces. Okay, these are nothing but the splenic sinusoids. Okay, next the cells which you see over here, actually it is a low magnification image, that's why you can't see the cells, but the cells are present in the form of cords, and these are called as splenic cords of Billroth. Okay, now can you identify? So see, this is the red pulp, this is the red pulp, this is the splenic cords in the red pulp, and this is the white pulp. See the eccentric arteriole. Okay, this is the eccentric arteriole. Here also you can see eccentric arteriole, and this is the capsule. So identification of the spleen under microscope is very easy. But क्या होता है, if you see the slide of the spleen and if you see the slide of lymph node, okay, some confusions are there, I have seen. Okay, next when we'll discuss about the lymph node, at that time we'll notice that appearance are almost similar under microscope. यहाँ पे नहीं, under microscope. Student used to get confused. Okay, so how to deal with that we'll discuss in lymph node part. Okay, so that is all about the spleen. I know most of the student have written this part. ठीक है, तो जितना लिखा that is more than enough. You don't have to write anything extra in the spleen. Acta. This one. Only this one. Okay. Next is the lymph node. Okay. Have you seen the macroscopic structure of lymph node? Anyone? In dissection, or have you seen the lymph node? Not देखा है lymph node? Anyone? In dissection hall, have you seen the lymph node? That day, while dissecting the head and neck portion, I got some lymph nodes. ठीक है, मुझे मिला था lymph nodes, deep cervical lymph nodes. Okay, तो पता नहीं अभी रहेगा कि नहीं. अगर रहता है तो I can show the lymph nodes. Okay. By the way, have you seen the macroscopic structure, not microscopic? If anyone asks you what is the shape of the lymph node, दिखता कैसा है lymph node? What is the shape है? No. ठीक है. If you see the shape of the lymph node, it is kidney shaped. Let me show you the diagram. So in practical notebook, this diagram is compulsory. You have to draw this diagram. ठीक है. Now this diagram is showing the macroscopic structure of lymph node. ठीक है. क्या दिखाई दे रहा है उसमें? First of all, what is the shape of the lymph node? Kidney shaped. ठीक है. Then you can see it is having a convex surface and it is having a concave surface. Now in the concave surface you are getting the hilum of the lymph node, same like the kidney. ठीक है, ये देखो, this is the hilum. Through the hilum what happens? Blood vessels are entering the lymph node, as you can see in the diagram. ठीक है, ये देखो, blood vessels are entering the lymph node, and what is coming out from the lymph node? That is the efferent lymphatic vessel is coming out from the lymph node. तो जो lymph node होता है actually it is receiving the efferent lymphatic vessels. ये देखो, जो convex surface होता है, the convex surface receives the efferent lymphatic vessels. Through that efferent lymphatic vessels, lymph is entering the lymph node. Okay, as you can see, the lymph is entering the lymph node. ठीक है. Now same like the spleen, lymph node is also covered by connective tissue capsule, as you can see over here. ठीक है. Then the trabeculae or septa are there, which is dividing into small lobules. ठीक है, ये देखो, this is the trabeculae. So this is one lobule, this is one lobule, this is one lobule. Right. Next, if you take one particular lobule, then it is divided into three parts. One particular lobule divided into three parts. So suppose you have taken one lobule, it is divided into three parts. First, this is the outer cortex, then this is called as paracortex, and this is called as medulla. Outer cortex, paracortex, and medulla. These are the three parts. ठीक है. Next, three different types of sinuses are present inside the lymph node. Three different types of sinuses. Now what are those three different types of sinuses? जो कैप्सूल होता है, ठीक, कैप्सूल के नीचे. In case of lymph node, okay, in case of lymph node, as I said, the convex surface is there. Now this convex surface, as you can see, the efferent lymphatic vessels is entering the convex surface. Through the efferent lymphatic vessel, the lymph is coming inside the lymph node. Next, as I said, it is having a connective tissue capsule and trabeculae. Now, what is the function of the trabeculae? It will divide the lymph node into small lobules. As you can see, this is one lobule, this is one lobule, this is one lobule. Now, one lobule, how many parts are there? 3 parts. What are they? Outer cortex, paracortex and medulla. 3 part रहता है, ठीक है? Next, in the lymph node, 3 different types of sinuses are there. What are those 3 different types of sinuses? First sinus, the sinus which is situated just below the capsule, that is called as subcapsular sinus. ठीक है, ये देखो कैप्सूल के नीचे यहाँ पे रहेगा सबकैप्सुलर साइनस, यहाँ पे रहेगा सबकैप्सुलर साइनस. नेक्स्ट, ये जो ट्रेबेकुले है, अराउंड द ट्रेबेकुले द साइनस which is present, that is called as trabecular sinus. और and in the medulla, huge amount or large number of sinuses are present, and these are called as medullary sinuses. Okay, these are called as medullary sinuses. Now, why these sinuses are present? Any idea? इतना सारा साइनस क्यों रहता है? Because of no, it will not store lymph. Lymph continuously circulate होता है. Lymph cannot be stored. Okay, because see, यहाँ से देखो lymph घुसता है, यहाँ से निकल के जाता है, तो it cannot be stored. Huh? No. RBC inside lymph node will get separated. Nah, spleen nah. This is lymph node. Now you are in lymph node. Spleen is done. The answer is very simple, and because of the transportation of the lymph. See, I have shown you from here it is entering, and then it is coming out. So through which direction? How it will go? How it will circulate, how it will travel through the lymph node? Through these sinuses. ठीक है, तो जो lymph होता है पहले it will go into the subcapsular sinus, then trabecular sinus, and then medulla, and from medulla through efferent lymphatics it will come out. Okay, and those sinuses together, they are called as channel. Through those channels the lymph is flowing, and those are lined by endothelium. Okay, those are lined by endothelial cells. Now, ये जो flow होता है lymph का, once the lymph is entering and then it is flowing like this and it is coming out, okay, the lymph node will do some filtration of the lymph. ठीक है, for example, if any pathogen is there or if any foreign material is there in the lymph, so it will be eliminated by the lymph node. Next, when it is flowing through the lymph node, more amount of lymphocytes are added in the lymph node. Okay, which can give different types of immune support wherever it is required. Okay, so those are some important functions done by the lymph node. Okay, now here you can see the microscopic structure, the overall structure. So first of all, see, same story is repeated. Here also you can see the capsule is present, which is made up of dense connective tissue. From capsule, what is extending into the parenchyma? Trabeculae. ठीक है. Next, if you see the parenchyma, as I said, it is made up of cortex and medulla. But cortex, 2 parts of cortex are there: outer cortex and paracortex, or also called as deep cortex. ठीक है. Now, what is the appearance of the cortex? It is darkly stained. ठीक है. So always keep that in mind. जितना भी lymphatic organs होता है, अगर उसका cortex है, then cortex is darkly stained. Now, in this case, why it is darkly stained? Because large number of aggregation of lymphocytes are present. Okay. Now, 2 parts are there of the cortex. What are they? These are the outer cortex and paracortex. Now, in case of outer cortex, what you will be getting? It is nodular in appearance. Why? Because of the presence of lymphatic nodules. Okay. The lymphatic nodules are present in the outer cortex. That's why it is nodular in appearance. And in some cases germinal center will be there, in some cases germinal center will be not there. Okay, and you can see B lymphocytes are present in the outer cortex. Next, the paracortex lies between the outer cortex and the medulla. Now it contains T cells predominantly, that's why it is called as thymus dependent zone. ठीक है, जो आउटर कॉर्टेक्स और मेडुला के बीच में जो कॉर्टेक्स है, दैट इज कॉल्ड एज पैराकॉर्टेक्स. इट इज ऑल्सो कॉल्ड एज थाइमस डिपेंडेंट जोन. व्हाई? बिकॉज मोर नंबर ऑफ टी लिम्फोसाइट्स आर प्रेजेंट. ठीक है, दैट्स इट. Next, if you see the medulla, first of all, it is lightly stained. Why it is lightly stained? Because the concentration of B lymphocytes are less. Okay. Now these B lymphocytes are mainly in the form of cords, and they are separated by medullary sinuses. Huge amount of sinuses are present. So when you will see the slide of spleen and when you will see the slide of lymph node, okay, the best way to differentiate is when you will focus on the medulla, you will see so many spaces are there, large spaces. Those are nothing but the medullary sinuses. Okay. Next, तो everywhere don't forget to mention the reticular meshwork. कोई भी lymphatic structure हो, उसका जो framework होता है, meshwork होता है, that is formed by the reticular cells, reticular fibers. Okay, and in this case we are getting some dendritic cells. Dendritic cells are nothing but the antigen presenting cells. Okay. Next, sinuses. So how many types of sinuses are there? 3 types of sinuses. First, you can see subcapsular sinus beneath the capsule, then trabecular sinus adjacent to the trabeculae, and medullary sinus where they are present in the medulla. Okay. So as you can see, the lymph which is entering, first they will go into the subcapsular sinus. From there it will reach the trabecular sinus. From trabecular sinus it will go to the medullary sinus, and then it will come out through efferent vessel. Okay, this is the direction of the flow of lymph inside the lymph node. So all those things we have discussed, just go through it, you will understand. So can you identify the parts? पहले क्या देखना है? First, what you have to see? Capsule. Very good. Can you see the capsule? ये देखो, this is the capsule. Below the capsule, can you see the subcapsular sinus? ये देखो, this is the subcapsular sinus. After that, can you find out the trabeculae? Where is the trabeculae? See, this is the trabeculae. Okay. Adjacent to the trabeculae, you can see trabecular sinus is also there. Okay. Now, which part is the cortex? Outer part will be the cortex. As simple as this. So, ये पूरा part cortex है. ये पूरा part. Subcapsular sinus के नीचे से. See, this is the capsule. This is the subcapsular sinus. So, from here to here, ये पूरा cortex है. ठीक है. Now, cortex में कितना part रहता है? Two parts are there in the cortex. So, this entire part is the outer cortex, which is nodular in nature. Why it is nodular in nature? Aggregation of lymphatic follicles are there. So, see, this is one lymphatic follicle, and here you can see the germinal center, with or without germinal center. Okay. Next, this particular part is the paracortex. As you can see, this particular part is the paracortex. Okay. So, paracortex is also called as Thymus dependent zone. Why? More number of T lymphocytes are present. ठीक है. यहाँ पे रहेगा B lymphocytes. Okay, यहाँ पे रहेगा T lymphocytes. ठीक है. Deep to the cortex, which part is this? Medulla. Medulla में क्या रहता है? Yes, medullary sinuses are there, but along with that B lymphocytes are present, and they are in the form of cords separated by sinuses, and these are called as medullary sinuses. Okay, so the direction is like this, the flow of lymph. Here you can see, so this is a low magnification image of lymph node. Okay, low magnification. Can you identify the capsule over here? यहाँ पे can you identify the capsule? You can't see the capsule. ये देखो, ये this is the capsule. ये वाला capsule है. Then this is the subcapsular sinus. ये दो subcapsular sinus. Can you see the lymphatic nodules? See, these are the lymphatic nodules. Okay. So the lymphatic nodules are present. Next, here you can see, this is the medulla. So this particular part will be the paracortex or thymus dependent zone. Okay. So only these things you have to identify. Now this is the high magnification image of lymph node. ठीक है. इसमें भी देखो, ये capsule दिखाई दे रहा है. Subcapsular sinus. ठीक है. Then you can see here also you can see the subcapsular sinus. Here you can see the trabeculae. This is also a beautiful diagram where all the parts can be seen of lymph node. So lymph node में, whenever you see the lymph node, two things you have to focus. First, see the subcapsular sinus, and see the sinuses in the medulla. It will be easier for you to identify. ठीक है. Okay, next, have you seen the thymus? Anyone? Have you seen the thymus in dissection hall? Thymus दिखाई देता है कि नहीं? हाँ? What is the location of the thymus? Lung? हाँ, behind sternum. Which part of the mediastinum? Thymus. हाँ, superior mediastinum. So you have dissected the superior mediastinum. Have you seen the thymus? Why? No. Is? No. That is the reason. It degenerates. Okay, the thymus just after birth, the size start increasing up to puberty. It reaches its maximum size, and after that it starts degenerating. Okay, and then you will be getting thymus as adipose tissue. ठीक है, इसीलिए जब डिसेक्शन किया थाइमस दिखाई नहीं दिया. ठीक है, तो thymus and bone marrow, these are primary lymphoid organs. Okay, these are primary lymphoid organs. Now if you see the thymus, it is bilobe structure located at the anterior part of superior mediastinum behind the sternum. Okay, how it develops? It develops from third pharyngeal pouch, and mesoderm contributes the thymocytes and connective tissue. Now here you can see at birth the weight is 10 to 15 gram. ठीक है, weight increases 30 to 40 gram at puberty, and then it is replaced by fat. That's why you have not seen the thymus when the dissection was done. ठीक है, and thymus is also having rich blood supply. Now you have to understand the structure of the thymus. ठीक है, so what present in the thymus? So here also you can see connective tissue capsule is there. ठीक है, now one particular point which is related to the connective tissue capsule is the septa or the trabeculae, which is extending into the thymus. They are incomplete. Okay, they are incomplete. That's why the lobules which are formed, they are also incomplete. Okay, here you can see the septa divides the thymus into incomplete lobules. And if you take one incomplete lobule, okay, it is also having cortex and it is having medulla. So one incomplete lobule, another incomplete lobule, the medulla is continuous with each other. This is one important feature, identifying feature of thymus. ठीक है, next parenchyma में दो part रहता है, cortex and medulla. Here also you can see cortex darkly stained, medulla lightly stained. Very simple. ठीक है, तो in case of cortex, closely packed T lymphocytes are there. Why it is darkly stained? Because the aggregation of cells are compact, okay, and more amount of T lymphocytes are present. That's why the cortex is darkly stained. Medulla, it is lightly stained because loosely packed T lymphocytes are there. Along with that, another important feature which is present in medulla, that is the Husserl's corpuscle. Have you heard about it, Husserl's corpuscle? It is a unique identifying feature of medulla of the thymus. ठीक है, what is the appearance? Hyaline material. Hyaline material doesn't mean that hyaline cartilage. Okay, the appearance is hyaline material. ठीक है, which is surrounded by type 6 thymic epithelial cell. Now I am coming into what is thymic epithelial cell. Okay, or also called as epithelio-reticular cells. Okay, so this is the medulla. Now another important point in the thymus is there are 6 different types of epithelio-reticular cells. Okay, 6 different types of epithelio-reticular cells are present in the thymus, and they are having very important function. ठीक है, तो ये जो epithelio-reticular cells है, these are also called as thymic epithelial cells, specialized cells which are present in the supporting role in thymus. Okay, for example, this 6 types of cells are there, right? तो उसमें से 3 types of cells are present in the cortex and 3 types of cells are present in the medulla. Starting from the maturation of T lymphocytes, producing the barrier, blood thymic barrier, okay, producing the junction between the cortex and the medulla. Okay, all the functions are done by these epithelio-reticular cells. Okay, that's why these cells are extremely important. Now, can anyone tell me why blood thymic barrier is there? Thymus is producing T lymphocytes. So nothing to do with the antibodies. Any idea why blood thymic barrier is present? And it is very important. We have to study about the structure of blood thymic barrier. For that, you have to understand the differences between primary and secondary lymphoid organs. If that concept is clear, then you will understand this part. Think. I will give you the answer at the end of this class. Think what is the importance, why it is required to have blood thymic barrier. Okay. So in case of thymus, as you can see, consists of right and left lobes joined by a fibrous tissue. Connective tissue capsule is there. Septa will go and divide in incomplete lobules. Here you can see the diagram. So this is the connective tissue capsule. Okay. From the connective tissue capsule, you can see the septa is going and divided into incomplete lobules. So, for example, this is one lobule, this is one lobule. Now, these are incomplete. This is cortex, this is medulla. Medulla is continuous with the adjacent lobule. Okay, this is one important identifying feature of thymus. Okay. Next, if you see cortex, it is divided into two parts, cortex and medulla. First of all, cortex is darkly stained. Medulla is lightly stained. So here you can see cortex is darker. Okay, closely packed T lymphocytes are there, and meshwork of epithelio-reticular cells are present. Very important. As I said, 6 types of epithelio-reticular cells are present. Okay. Next, in the medulla, lightly stained. Okay. It also having epithelio-reticular cells, and along with that, Husserl's corpuscles are present. Okay. Now, can anyone tell me why these Husserl corpuscles are present, and what is the importance of these Husserl's corpuscles? So, first of all, Husserl's corpuscles are present only in the medulla of the thymus. Okay. Now, these are round ovoid structure present in the medulla, consist of eosinophilic hyaline mass. So, what will be the appearance? Eosinophilic appearance. मतलब कैसे होगा? Pink in color. Okay, eosinophilic rounded hyaline mass, which is covered by type 6 epithelio-reticular cells. Okay, and these epithelio-reticular cells which are there type 6, they are secreting IL-4 and IL-6. IL क्या है? Yes, interleukins, interleukin 4 and interleukin 6. Okay, which are signaling protein for any kind of inflammation and infection in the body. Okay. Next, here you can see the list of the 6 types of epithelio-reticular cells. Okay, 3 present in the cortex, 3 present in the medulla. So if you see the type 1, type 1, mainly they are forming the blood thymic barrier. Okay, the blood thymic barrier, I am coming into the blood thymic barrier that is formed by the type 1. If you see the type 2, it is isolating the compartment for development of T cells. Okay, that means T lymphocytes, helps in development of the T lymphocytes. Now type 3, it forms the functional barrier between the cortex and the medulla. And if you see type 4, they adjacent to the type 3, along with type 3 they form the corticomedullary junction. Next, from here onwards, all the cells are present in the medulla. 5 cellular framework of the medulla, and 6, as we have seen around the Husserl's corpuscles, type 6 cells are present, and they secrete IL-6 and IL-4. Okay, helps in the thymocyte maturation. So these are the 6 different types of epithelio-reticular cells, or also called as thymic epithelial cells. Okay. Next is the blood thymic barrier. So if any barrier is there, how to study the barrier? Today in the morning class I have mentioned layers. Okay, कोई भी barrier हो, you have to know the layers that sit. Okay, either from inside to outside or outside to inside. Anyway, ठीक है. So here you can see the diagram of the blood thymic barrier. ठीक है. So first of all, you can see this is the blood vessel. Okay, this is the capillary wall. तो capillary wall में endothelium रहता है. This is the capillary endothelium. Now this is the basement membrane of the capillary endothelium. ठीक है. After that, here a special type of cells are present. These are called as pericytes. Okay, ये देखो, these are called as pericytes. which will control the flow of blood through the capillary bed. That's why pericyte cells are present. Next, on the outer aspect, you can see this particular part. Okay, this is the perivascular connective tissue. This particular part is the perivascular connective tissue, and then outside to this again a basal lamina is there. Now this basal lamina is for the type 1 reticuloepithelial cells. Okay, epithelial reticular cells. उसका जो basal lamina है, this is the basal lamina, and outside that you can see immature T lymphocytes are there. ठीक है, तो how the layers are formed? So this is the innermost part of the layer. This is the outermost part of the layer. Okay. Now on the outermost part you can see the T lymphocytes are developing. Now ये जो barrier है क्यों रहता है? T lymphocytes here they are immature T lymphocytes. They are developing. ठीक है, तो blood में अगर मान लो कोई antigen है, this T lymphocyte should not get exposed to that antigen. If it is exposed to that antigen, इसका development रुक जाएगा. The development will not take place properly. Okay, that's why the barrier is given. Next, can anyone tell me some hormones secreted by thymus? Then, yes, thymopoietin alpha. Okay, so many hormones are there. Okay, before going to the hormones, this is the microscopic structure of thymus. What you can see in the diagram? No. First, you don't have to identify the hustle. First, what you have to identify? First you have to identify the capsule. Then you have to identify the blood supply. Yes, incomplete septum or trabeculae. So here you can see these are the incomplete septa or the trabeculae. Okay. Now if you focus over here, ये देखो, this is one incomplete lobule, this is one incomplete lobule. ठीक है. Now in this incomplete lobule, this is the cortex. This is the cortex. ठीक है. This is the medulla. Now see, medulla is continuous with this medulla. ठीक है. Why? Because the septa is incomplete. Next, जो cortex होता है, it is darkly stained. Why it is darkly stained? Yes, because closely aggregated T lymphocytes are present. That's why it is darkly stained. Next, you can see the medulla. It is lightly stained because the aggregation is not closely placed. But in case of medulla, also T lymphocytes are there. Next, what is present in the medulla? Husserl's corpuscles. ये देखो, these are the Husserl's corpuscles. These are the Husserl's corpuscles. Okay. So, in order to see the Husserl's corpuscles clearly, what you have to do? You have to see the magnified view. Then only you can see the Husserl's corpuscles clearly. Okay. So, identification of the thymus under microscope is also simple. It's not that difficult. Now, here you can see magnified view of the thymus, where you can see the Husserl's corpuscle and you can see the type 6 reticuloepithelial cells, and then you can see the aggregation of lymphocyte. तो ये कौन सा पार्ट होगा? अगर Husserl's corpuscle से, then which part will be this? Medulla. See, this is another. This diagram you have to draw. Thymus. No one have written about the thymus. You have to write the thymus. And this one you have to draw in your practical notebook. ठीक है? What you can see over here? क्या दिखाई दे रहा है? First of all, you can see the medulla. Okay, then you can see the Husserl's corpuscle, you can see the cortex. Okay, then see, these are the incomplete septa, these are the incomplete septa. Okay, and see, medulla is continuous with each other. ये देखो, medulla continuous होता है with the adjacent incomplete lobules. Okay, and these are the list of hormones secreted by the thymus. Okay, thymulin, thymopoietin, thymosin alpha, thymosin beta 4, thymic humoral factor. These are the hormones secreted by the thymus. Okay, so that's all about the lymphoid tissue. Now I am having some slides, so you have to see the slides. Okay,